Okay. Oh, I forgot. I wanted to give you all this. So all I did was I took the two little tables that are in your textbook. So for those of you who are on Zoom, I took the two little tables of abbreviations. I was like, why is my folder so heavy today? And I put them together. So you have like one place to go and look for these abbreviations. Now, the first two columns are what I'm going to give you on the exam. I'm not giving you the function, but I'm giving you the first two columns, so the abbreviation and what that abbreviation means. So I'm going to go ahead and hand these out. I'll just give a couple for everybody. Figure out the extras. Yeah, just yeah. Figure out the extras. Yes, and if you're in my section and need to turn in a lab report, please turn in a lab report. Thank you. Awesome. Okay. I didn't really count how many were there. So if you don't have enough, look at the row on the side of you, and they should have enough for you. Okay. No, but I'll stay with you. Okay. Thank you. All right, y'all ready? So let me ask you a question. How do you terminate a GPCR signal? Happy Monday. How do you terminate a GPCR signal? So the one way to do it is called the G protein cycle. Right? Yes. Oh no, I didn't do a joke. Thank you. Let me remember. What does a whale fight with? A swordfish. Some days are better than other days. Here, I got another one for you because that one was kind of lame. What do you call an alligator in a vest? An investigator. Okay. So instead of having one bad joke, you had two today. All right, so G protein cycle. Do not look at the slide. Try not to look at the slide. And tell me what happens in the G protein cycle. I want you to start with receptor, ligand binds to receptor. So ligand binds to the GPCR. Then what happens? What happens? What does that mean? Turns it on. It exchanges what for what? So it exchanges our GDP exchange to GTP, right? And this is usually on G alpha. And what makes that happen? Well, ligand binding to the GPCR, and what do we call something that changes a nucleotide in that way? Guanine exchange factor. So there's inherent guanine exchange factor ability in the GPCR that allows for that exchange to happen, right? Okay, so now what? Well, so that's actually not two. That's one way. This is really, we'll call this A, step A, right? Then step B, what happens? So now we have G alpha bound to GTP. What happens? There's a lot of things that happen between those two things. Subunits dissociate. So we have G protein subunits dissociate. So I have a G protein alpha disassociating, and I have a G protein beta gamma, right? Now that they're disassociated, they can induce signaling, right? So yes, we could go into extensive detail about that, but that's not what this is about today, right now, right? So we're just going to say downstream signaling. And this is, again, you'll have some other downstream signaling, right? We're just going to say that that's going to happen, because what are we talking about right now? We're talking about terminating the signal, right? Okay. So what happens next? I want to turn it off. Right now, what is it bound to? This G alpha, what is it bound to? GDP or GTP? GTP. When it's on, it's bound to GTP. So in order to turn it off, what do I need to do? Turn it back into GDP. So that's a intrinsic ability of the G alpha subunit, but it needs to be stimulated. What stimulates that? A GAP. Guanine, what? Look on your sheet. What is GAP? In the top box. GTP-ACE activating protein. So what is a GTP-ACE activating protein going to do? It's going to stimulate, right, intrinsic GTP-ACE activity of G alpha. Okay. So what happens? Now I have G alpha, and is it bound to GTP or GDP? Now it's bound to GDP, right? Because I hydrolyzed GTP to GDP. So what did I do? I just hydrolyzed a phosphate group. Okay, now what? Yes, reassociation. So G alpha now in the GDP form. So you put GDP form, reassociates, right? Because we want to be primed and ready for the next signal with G beta gamma, right? And that's going to turn off G beta gamma. So now everything is off again. And then reassociates with G beta gamma and the GPCR. And we back to where we started. So if there is a figure that we talk about in class, if you can do this, this is a good study tool, right? You get out a dry erase board, you get out a sheet of paper, and you're like, okay, I'm going to describe the G protein cycle. Don't look at your figure in the book. Explain it. Explain it to the dog. Explain it to your plant. Explain it to whoever. It doesn't matter. If you have a study group, you could take turns, but this would be a really good thing to do. Okay. So this is not the only way that you can terminate a GPCR signal. This is one of the two ways that you can terminate a GPCR signal. The other way that you can terminate a GPCR signal is through desensitization after the subunits have disassociated. And you can actually shut down the receptor. So you either sequester the receptor or you degrade it. Either way. Either it's in a vesicle and it's hidden, or you actually degrade it. And so how does this work? Well, there are kinases. There are kinases for everything, right? That's what you've learned in the last couple of weeks. There are kinases for everything, right? So the kinases will phosphorylate your GPCR receptor. And it'll phosphorylate it on the cytoplasmic side, right, on serine and threonine residues. And what that does is it starts a cycle that will eventually either sequester the receptor or degrade the receptor. So you have, right, your ligand that's going to bind to your receptor. When that happens, right, all the activation steps that we talked about actually occur. But after that activation occurs, the beta gamma subunit, the dissociated, remember that was one of the steps that you now had a G protein alpha subunit and then a G protein beta gamma subunit. Remember that from, where was it? Here, see this? And it initiates downstream signaling. So that's target signaling and some other things that can happen. So one of the other things that can happen is that this beta gamma is going to bind to something called, we call it for short BARC, right? But what, go and look on your sheet. What is BARC? For those of you who came in, there should be some extra sheets in the back somewhere. For those who came in late, if there are any in the back, would y'all pass them over or something? So all we're doing is we're looking at our abbreviations. So what is BARC? That they're kinase. So it is a kinase that's going to phosphorylate the receptor, right? So we're going to recruit this kinase, and this kinase is going to go in and actually phosphorylate the cytoplasmic part of the receptor. Once that cytoplasmic portion of the receptor is phosphorylated, then you can actually recruit some other things. So like PKA. PKA is another kinase, protein kinase A. Those can go in and phosphorylate our cytoplasmic tail of our GPCR, right? So now we have this GPCR that's phosphorylated all over the place, right? That cytoplasmic tail, do you see it has all those phosphate groups on it? Okay, it's all phosphorylated. If it's phosphorylated, this is a signal to this protein called beta-arrestin. Beta-arrestin is going to come in and bind to the receptor, but it only binds to the phosphorylated tail of the receptor. So when that beta-arrestin forms, now we have this complex, and the cell is going to internalize this whole complex. And now the receptor is stuck in an endocytic vesicle. Is it going to be able to bind ligand? No, it's inside the cell. It's sequestered. So sometimes it hangs out there, and sometimes it gets degraded. So sometimes you want to get rid of it completely, and so the cell will degrade it, and sometimes you want to recycle it back to the membrane. So if it becomes dephosphorylated, it can recycle it back to the membrane, but otherwise it's degraded. So it just kind of depends on which signaling mechanism is working, whether it's degraded or not. So could you write yourself like something like this? What we just did, what we just did here. How do you terminate a GPCR signal? 1 is the G protein cycle. 2 is what? Receptor desensitization. I can't even say it. Desensitization. I'll spell it, right? And then you could go through and you could do that pathway, right? So I'm trying to give you all some tools on how to study. Okay. All right. So we just couldn't quite finish that in the last class. I guess I went off on my side trip too much, but that's okay. So today we're going to really focus on the second major class of receptors, and those are receptor tyrosine kinases. So what do you think these receptors are going to intrinsically be able to do? Phosphorylate tyrosines. And they're going to actually autophosphorylate. And we'll talk about exactly what that is. So these are going to bind to whatever ligand that's exterior from the cell, and it's going to activate the intrinsic tyrosine kinase function of the receptor itself. So the receptor has kinase activity, which is not what we see in a GPCR, right? In a GPCR, when our G proteins dissociate, those activate kinases. But here, our receptor has its own kinase activity. And so these class of receptors typically function as dimers. So they're separate when they're not bound to ligand. When they bind ligand, they dimerize. So we're going to talk about all the downstream signaling proteins that then bind to our phosphorylated receptor. So what is the receptor tyrosine kinase? Once it's phosphorylated, what is it going to do? Well, it's going to phosphorylate things, or it's going to associate with things that will recognize the phosphorylated groups on the receptor. So we call the next proteins in the cascade, we call them adapter proteins. So adapter proteins, you can put over here, right? These are kinases. So adapter proteins, these are kinases, right? So they will bind to the phosphorylated receptor and they will go on and phosphorylate other targets. So what we're going to see today is a phosphorylation cascade over and over and over again. So eventually, right, all this kind of stuff is going to happen due to a phosphorylation cascade. So what are some examples of receptors that function in this way? And those are the epidermal growth factor receptors and the insulin receptor. So when we talk about epidermal growth factor receptor, a lot of the ways that we recognize and have discovered these receptors is through mutations. So for example, if you mutate the epidermal growth factor and it is always on, what do you think you're going to have? Growth, right? What do we call that when it's continuing and it spreads? We call that cancer. We call that cancer, right? So mutations that turn the EGFR receptor on lead to cancer. How about insulin? What if we turn the insulin receptor off? What do you have? Diabetes, right? So a lot of the discoveries we make are through mutations. That's a really interesting way to think about things, right? Okay, so here's kind of our diagram. Usually when it's inactive, we have a monomer. So this is our receptor tyrosine kinase in the membrane as a monomer. One of the two will initially bind a ligand. That will induce dimerization when both of them bind to the ligand. Then what is going to happen is you're going to have activation of the intrinsic kinase activity in the cytoplasmic portion, right? So kinase activation, I would add that in cytoplasmic tail, right? So the kinase activity is here. So what happens is that you're going to have phosphorylation of both cytoplasmic tails, right? They're going to phosphorylate each other. Now that they're phosphorylated, now there are perfect sites for attachment of those target proteins. So all of these different proteins are adapter proteins, right? And these adapter proteins, do you see this conversion of ATP to ADP? What does that tell you that this red one can do? It can use energy and phosphorylate something, right? Now this phosphorylated adapter protein can go on and activate something else, which can go on and on and on and on, right? And that's what we're going to talk about today. What is this cascade and how does it work, right? So the other thing is that the cytoplasmic tail itself can phosphorylate a target protein because it has intrinsic kinase activity. So not only can its phosphorylated groups recruit things, but it can in and of itself phosphorylate a target protein. So there are going to be multiple pathways here. But in the end, right, we're going to activate huge phosphorylation cascades. So let's talk in particular about one. We're going to talk about the EGFR, right? Epidermal growth factor is the serum. Epidermal growth factor receptor is the receptor, right? So the serum growth factor binds to the receptor and it stimulates dimerization, right? We already talked about that. So what we see is a two-step model. If you have these two receptors, right? Ligand is going to bind to one receptor first. Doesn't matter which one, but it's going to bind to one first. That initial binding is going to induce the dimerization. That induction of dimerization is going to lead to kinase activity of one receptor phosphorylating the tyrosine residues on the other receptor. So one of them does it to the other. Now, once that second receptor is phosphorylated, it becomes active. And it can then go in and phosphorylate the first receptor. So we see it in a stepwise fashion. And each of these events, ligand binding, dimerization, phosphorylation of the first phosphorylation of the second receptor, then phosphorylation of the first receptor, all lead to conformational changes that are required for the next step. And you can see that when you look at crystal structures and things like that. So let's look at what that looks like. Normally, they're single, right? They're by themselves. And what you'll see is they have a extracellular portion that's going to bind the ligand, transmembrane portion, and then you have a cytoplasmic portion. But we divide the cytoplasmic portion kind of into two areas. One of the areas, these little circles, these are places where the receptor can be phosphorylated. And then you also have an enzymatic activity portion of the C terminus, right? So we have enzymatic activity in one part, and then we have sites that need to be phosphorylated in the other. So when our ligands come in and bind, and they'll bind sequentially, first you'll have one ligand bind, then you'll have the second ligand bind. But that ligand binding will actually induce dimerization. So you actually have first those cytoplasmic tails associating with one another. And so whichever one bound ligand first is going to phosphorylate the cytoplasmic tail of the other, right? And then once this one's phosphorylated, so you see it turning yellow. See them turning yellow here? That means they're phosphorylated. So green is unphosphorylated, yellow is phosphorylated. So once these tyrosine residues are phosphorylated, then they can start to bind to other ligands. So we have one receptor phosphorylating the others. I guess it's this one. This one's phosphorylating this one's tyrosine residues. Then once this one is all phosphorylated, all yellow, it'll start phosphorylating the first receptors, tyrosines. So now we have these phosphotyrosines that are ready to recognize these adapter proteins. And those adapter proteins will recognize those phosphotyrosine clusters, and we call them SH2 binding domains or SH2 binding domain proteins, right? These contain the phosphotyrosines. That's the PY, phosphotyrosines. Okay. So now, what happens after that, right? Now we got to talk about some signaling cascades. So we have our activated receptor. It's already dimerized. We've already phosphorylated those cytoplasmic tails. Now what happens? The first thing that we need is an adapter protein. In the EGFR mechanism, that adapter protein is called GRB2. So go on your sheet, go find GRB2. Where is GRB2? What is GRB2? So this is a protein that will bind to the growth factor receptor, right? So I know some of these names sound kind of weird, but when you go and you look at the name, they actually sort of named it okay. Some of them, I won't say all of them, but some of them, right? Okay, now this adapter protein is going to link the receptor to our downstream signaling events, right? So what we want to do is to activate that cascade. So we're going to link to a protein that has some kind of enzymatic activity, right? So the first protein, the next protein in the cascade is called It's called SOS, which stands for Son of Sevenless, which is a ridiculous name, but it was found in Drosophila, and the naming scheme in Drosophila is ridiculous. It's absolutely ridiculous. They wanted to have fun, and it makes no sense. Yeah, they have so like cytoskeletal proteins that are named like Skeletor and like ridiculous things. You know, so the point is that this is a GEF protein. What's a GEF? Guanine exchange factor. A guanine exchange factor. If you have a guanine exchange factor, what can that do? That can take a G protein that's bound to a GDP and make it GTP. Now we can activate that same set of downstream signaling events that occur when you have a G protein that's associated to GTP. So there are some similarities and there are some differences in the pathways. This side of the pathway gives you very much similarities to GPCRs, right? Because we activate this G protein is called RAS. Now, do you want RAS to be on all the time? Remember, this is a growth factor receptor. So growth factor tells things to grow. No, when you're a kid, right, or you're a developing fetus, something like that, yes, you want things to grow. But we want to be able to turn off the signal. Please make sure y'all masks are on for me. So you want to be able to turn this signaling off. So what happens is we have a RAS gap. What's a gap? So look on your sheet. GTPase activating protein. So this will promote RAS's intrinsic GTPase activity so that RAS will get recycled back to the GDP form. That's really important. What happens if you were to have a mutation in RAS that prevented its association with RAS gap? What if you didn't have this association? You have uncontrolled cell growth. This is about 80% of pancreatic cancers have RAS mutations in them. This is one of the most common proteins that is mutated in cancer, right? Have y'all heard of the difference between oncogenes and tumor suppressor genes? Have y'all heard that before? Some people, yes. So think about it like you're driving a car. This is how I like to think about it, right? If I have something that's an oncogene, I only need one copy and I'm going to have cancer because that gene is a gain of function. That mutation will result in a gain of function. So it's like I'm driving the car and I'm hitting the gas, right? Doesn't matter if I only have 1 foot or 2 feet hitting the gas. If you hit the gas, the cell's going to continue to divide and we're going to have cancer, right? You can have a mutation, that's an oncogene. A tumor suppressor gene is a gene that is normally there to stop cell growth, right? So usually something in the like cell cycle check or something like that. And so if you have one copy that gets mutated, your other copy will still stop the car. It'll still press the brakes. But if you lose them both, you have nobody pressing the brakes anymore and your car keeps going, right? So the car keeping going is turning into cancer. If you keep hitting the accelerator, that's an oncogene. If you stop hitting the brake, that's a tumor suppressor gene. Yeah, so you can get cancer a lot easier with an oncogene because usually you have tumor suppressor genes, you have multiple that do the same thing, right? So you have multiple stop gaps. Okay, so RAS is, like we said, a G protein. It's a G alpha protein and is anchored to the membrane. It's not going to go anywhere, even when it becomes activated. So we know it's activated by our guanine exchange factors, our GEFs. And we know that once it becomes activated, it's bound to GTP. To turn it off, we have to activate that intrinsic GTPase activity that's regulated by the guanine ATPase activating proteins. Whenever you see the abbreviation, if you think in your mind the name, it will help you to get more comfortable with the abbreviation. But I will be giving you this on the exam, right? But which portions am I giving you? The abbreviations and the names. I'm not giving you the functions. Okay, so be careful with that. All right, so how does this work? Let me zoom. I really want to zoom in on this. Okay, so, right, once RAS becomes activated, what does RAS do? Well, it's going to be a very similar phosphorylation cascade, right? So we have GTP bound RAS. That means that RAS is active. It is going to bind what's called RAF. So RAF is going to, once it's associated with RAS, it is going to be phosphorylated by SRC. SRC is a kinase, right? So we're going to see like all of this kinase activity. So now that RAF is phosphorylated and active, it is going to go and phosphorylate another kinase. That kinase is MEK. They have to be abbreviations for everything, right? So MEK becomes phosphorylated, activates another kinase. This kinase becomes phosphorylated. It goes to the nucleus. Once ERK is phosphorylated, now it can go into the nucleus, and it is actually going to phosphorylate transcription factors, right? What do transcription factors regulate? Gene regulation, right? So you have to have certain genes on and off if you want your cells to replicate or not. So we call these three kinases, RAF, MEK, and ERK. These three. These are MAP kinases. So we call it the MAP kinase cascade. The MAP kinases are mitogen activated proteins, right? So what's a mitogen? Mitogen sounds really familiar to mitosis, right? So these are things that will promote mitosis. So kinases that participate in the MAP kinase pathway will increase cell division. So what happens if you get mutations in those pathways? You are going to disrupt cellular replication. Maybe you downregulate it, maybe you upregulate it. It depends on the mutation, right? So have y'all seen some of these pathways before in physiology? Yes. Not like this.
Like, you see like epinephrine or acetylcholine.
Uh-huh.
Or like hormones.
Those kind of things. Okay. So they're similar pathways. They're similar in that you're going to see all of these phosphorylation cascades, right? Okay. So let's do full screen. Okay. So remember how we talked about if you mutate RAS, you're going to get cancer? So this is exactly that pathway, right? So even when you're not getting a ligand stimulating your receptors, if you have a mutant RAS, that mutant RAS is not going to exchange its GTP for GDP. So it's basically going to get stuck in that on position. It's always going to recruit RAF. It's always going to go down that MAP kinase pathway to promote cell proliferation. Even though your body's not making the hormones that are saying, hey, it's time to grow, you know, you're done growing, you're not supposed to be getting this signal anymore, and you're not, but RAS is mutated, and so it's going to promote this whole pathway, and you're just going to keep growing and growing and growing and growing, and that's cancer, right? So this is an example of an oncogene. Right, so it's a gain of function. Gain function. Okay. All right, so insulin, so y'all know a little bit about insulin, right? A little bit maybe. So the insulin receptor is similar, right? In that whenever you have this ligand bound, you're going to induce a conformational change inside of your receptor. But insulin's a little different in structure. The insulin receptor itself, so the insulin, this should be insulin receptor, right? Contains two chains. So you have an alpha chain, and this is another alpha. Then you have a beta and a beta. So you have two alphas, you have two betas. So you have an alpha 2, beta 2. So what do we call that? What do we call that kind of a protein? A what? So, well, you could talk about a homo or hetero, right? But it's two of the same with two different. So we just call it a tetrameric protein, right? Because you have actually how many subunits? 4. So you say it's an alpha 2, beta 2 tetrameric protein, right? Okay. So remember how we said if you have different subunits, when you're folding, when you get to that quaternary structure, what is one of the most common ways that we hold together those subunits? What kind of interactions? Side chain interactions got to be disulfide bonds going to be your strongest ones. It's not the only ones, but in this case, it is disulfide bonds. We're going to go through insulin receptor activation, but you should absolutely watch the animation. It's awesome. I always say that, but it's awesome. Okay. So when insulin binds, there are two different pathways that can happen, right? You can go down the pathway where we're going to activate GRB2, son of 7-less, RAS, right? Because when you eat, that tells your body that you have enough resources to grow, right? So you are going to activate expression of genes that control cell division. That's really important, right? You don't want to start growing when you haven't been eating. You can't waste resources on that. So one of the things when insulin levels is high is that we can grow our cells. The other thing that happens when we have high insulin is we want to tell our cells you can take in the glucose, and then if you have extra, what can you make? Glycogen, right? So see how this is going to be the opposite of what we talked about last class? What did we talk about last class? We talked about breakdown of glycogen. We talked about increasing gluconeogenesis, right? We talked about inhibiting glycogen synthesis. So now we're doing the opposite pathway. The opposite is that we want to promote glucose uptake. We want to promote glycogen synthesis. So we have a separate pathway that's going to allow both things to happen in response to insulin. So we have our insulin that binds to the extracellular portion of our receptor. And so what you have is an autophosphorylation event, right? Where one kinase activity of one phosphorylates the other, kinase activity of this one phosphorylates this one. And then we also said that this kinase active portion of the receptor can actually go in and also phosphorylate adaptor proteins. So in this case, this is the insulin responsive IRS, insulin receptor substrate. That's what it is, insulin receptor substrate. So you have this adaptor protein, right, that is going to bind to the phosphorylated receptor. Then that phosphorylated receptor that's active because it's bound to insulin is actually going to go in and phosphorylate that substrate. Now that it's phosphorylated, we're going to activate an enzyme called PI3 kinase, right? Then we're going to go through the PI3 kinase signaling proteins. We already talked about going through the MAP kinase protein, so I don't think we need to do that again, right? We really just need to talk about going through the PI3 kinase pathway. So you ready to do that? Sure. Yeah, sure, right. Why not? So we're really only talking about half of the things that happen when the insulin receptor gets activated now. So we have our insulin insulin receptor substrate that's bound, right? We're going to activate this whole green thing is PI3 kinase. This kinase will phosphorylate what's called PIP2. Why do we call it PIP2? What's associated? 2 phosphates. So it's phosphorylated in 2 places. So what this kinase is going to do is it's actually going to phosphorylate it in a 3rd location. Once this PIP3 is phosphorylated, right? So now I have 1, 2, 3 phosphate groups. We can recruit PDK1. PDK1 is going to go in and phosphorylate AKT. So AKT is also recruited because we have our 3rd phosphate group here. So because we have PIP3, we're going to recruit AKT, but it's the PDK1, it's the kinase that's actually going to phosphorylate the AKT. And then we're going to activate glucose uptake, glycogen synthesis. And if you want to know all about that, you should absolutely take Biochem 2 because it's really super interesting. And we'll go into detail on that. But the point is, what kind of a cascade is this? What's happening? It's another phosphorylation cascade. So do you see how phosphorylation turns things off and on in the cell all the time? We see that over and over and over and over again. So my suggestion is that you go over these pathways. Pick images that I've covered in class and grill each other or grill yourself with a dry erase board. How does this work? What happens? You can keep your little sheet next to you. That's kind of why I gave you one, so you can hold on to it and use it, okay? Because you will have that on the exam. Yeah? Questions? All right. Maybe we'll have time tomorrow to go through a couple of little practice things. See how much time we have. All right, so I told you we're not going to spend a whole lot of time on tumor necrosis factor receptor signaling, right? You got a couple of slides and that's it. So the interesting thing about tumor necrosis factor is that you have this one receptor that is going to stimulate 2 different opposing cellular responses. One is cell death and one is cell survival. And the thing that changes whether ligand binding to the receptor is going to induce cell death or induce cell survival is dependent upon what adapter proteins are present inside the cell. So if you have lots of pro-apoptotic, what is apoptosis? Programmed cell death. If you have a lot of proteins in there that are signaling, hey, the cell's not doing so red hot, then it's going to activate cell death. If you have a low level of pro-apoptotic and a high level of anti-apoptotic, the cell's going to survive. And this is really important because it allows cells to make a very quick decision. Should it stick around or should it not? Why is that really, really important? Yeah, especially cancer, right? Any kind of diseased state that a cell is in, you want it to apoptose very quickly. As soon as those negative factors are around, you want it to shut it down and kill itself, right? You don't want this bad cell to continue to grow because it's going to have some kind of disease phenotype. So you want that on-off switch to be very easy for the cell to figure out what to do. So the TNF alpha is a homotrimer. So how many do you need to create an active receptor? 3, right? So the receptor, we call it the TNF receptor. It binds tumor necrosis factor alpha, right? That's what the TNF stands for. And just like every other receptor, it induces a conformational change, every single one. And it induces the change in the cytoplasmic portion of the receptor, which we call the death domain. I kind of like the names on these. It's just a little something different, right? So what happens when TNF alpha binds and we have that conformational change? What it's actually going to do is it causes a disassociation of this protein called silencing of the death domain. So you have a disassociation of a silencing protein. Right, it does sound like a death metal band or something like that. It really does. If anyone starts a death metal band, you should absolutely go with that name. So once you disassociate the silencing complex, now that receptor can bind to certain downstream signaling things. So this is the normal state, right? It's unbound and it's bound to these silencing proteins. So the SOD proteins are the silencing of the death domain proteins. So this TNF alpha receptor is not signaling. When we bind to TNF alpha, we're now a trimer, which it really should have been, these should have been farther apart, but it's okay, we'll forgive the book, right? So when TNF alpha binds, we trimerize, we've lost our silencing of death domain proteins. Now we're going to bind to TRADD. So what does that stand for? Oh, I don't think I gave you this one. Did I not? I didn't give you this one. What is it? TNF alpha receptor associated death domain. So these are going to initiate that downstream signaling cascade from occurring. So when we talk about that cascade that's going to actually induce apoptosis, it's the caspase. The caspases. Why do we call them caspases? Y'all have heard of caspases before, right? Yes, no? No? So yeah, some people have. So caspases are a class of enzymes that work on two different amino acids. Can you guess which two? Yes, cytosine. And I heard it, aspartate. Right? So these are going to be enzymes that cleave at those two residues. So they can actually go in, and it's a cascade. There's one that activates, there's one that actually gets changed. Remember how we said there are some enzymes that aren't normally active and then they get cleaved and then they become active enzymes? Remember that? The caspases fall into that category. So when they're in their native state and they're not enzymatically active, we call them pro-caspases. And then when they're active, we call them caspases. So the pro-caspases begin the cleavage, right? They turn into caspases, then they go and cleave other pro-caspases and turn them into caspases. And eventually you get down to caspase 3, which is called the executioner. And this is the one that goes in and really degrades those key proteins that the cell needs to survive. And so when you degrade those, the cell undergoes apoptosis and it's toast.
Does the TRAAD both happen in the cell death and in the cell survival? That happens in both?
No. So it depends. It depends on if you have these pro-apoptotic proteins present or not. If they are not present, you will not initiate apoptosis. So this is assuming that you want to promote apoptosis. Yep. All right, and that's all you need to know on the caspases. Quick and easy, right? Oh, I think we might finish today. What time is it? We might finish today so we can do some things tomorrow so that you get all the content before the day before the exam. Isn't that awesome? All right, nuclear receptors. Nuclear receptors are the only ones that are not bound to a membrane, right? These are inside of the nucleus. These act as transcription factors to actually change gene expression. So their ligands pass through membranes, right? So if we look at these, what are things that can pass through membranes? Steroids. Steroids can go through membranes. There are different metabolites that can go through membranes. It all depends on their functional groups and those kinds of things, right? So if you think about this receptor, the receptor itself is a protein, correct? So proteins are controlled by expression levels. How much you transcribe that DNA into mRNA and you turn that mRNA into protein. So we call that expression. So it's going to be governed by cell-specific expression of nuclear receptors. Because do you want every cell in the body to respond to the same steroid? No, right? So each cell is going to express certain subsets of our genes. Same thing is true about the transcription factors. So they're not on every gene. Then we also talk about bioavailability of our ligands, right? Are we going to always express estrogen, for example? No, right? You have different levels of it at different times and in different males versus females, right? And then each of those transcription factors has a place on the genome where it acts. Is that location accessible to the transcription factor or not? And that is all about condensation of your genome, right? How condensed is it? Is it heterochromatin? Is it euchromatin, right? And so if it's accessible, then, and the ligand is there and the transcription factor is there, it's all going to work. But if any one of those three things doesn't work, even if you have the ligand present, you're not actually going to get an effect. Okay? So it depends on all three things. So remember before how we said that transcription factors work in pairs? Didn't we talk about this a little bit? Maybe. It's a Monday. It's hard to remember. So typically transcription factors work in pairs. They either work as a homodimer or they work as a heterodimer. So sometimes they're going to bind the same ligand. Sometimes they need to bind to different ligands. So each of these transcription factors cannot work by themselves, right? Typically, alone, they don't associate with any other co-regulatory proteins. But once the dimerization occurs, now it's actually going to recruit co-regulatory proteins that then promote transcription of a gene, right? So if you look at things that are homodimers, the ligand binding sites are typically close to one another. So do you see how they're a head-to-head association, the yellow ones, where the green and the purple are a head-to-tail association? So if you think about it, if you have a head-to-head association, it's two transcription factors that work the same way, right? They're homodimers. So they have to recognize the same sequence, but one is in one particular orientation and one is in the mirror image orientation. So if you look at the DNA sequence is going to be an inverted repeat. So here we're binding AGAACA, but if you want to know what this one's going to bind to, well, we need to look at the complement and we need to go in reverse. So we're going to go T, G, T, T, C, T, and that's what we have, T, G, T, T, C, T. Do you see how that's an inverted repeat? Okay, so now look at a homodimer. I'm sorry, a heterodimer. Heterodimers are usually going to bind to the same DNA sequence, but recruit different co-regulatory proteins. So we have exactly the same sequence that one will bind that the other binds. And so you can look at the different ones, and typically the steroid receptors tend to be these homodimers, whereas metabolite receptors tend to be heterodimers. So, and you don't have to know the consensus sequence. You don't have to know that. All right. So this is just what we've already talked about, right? That you have this inactive nuclear receptor, right? And you're going to bind to some kind of ligand. And I'm not particularly happy with this one because what else do you need? We need another transcription factor, right? Because they act as dimers, right? So I'm not particularly fond of that thing, but just remember we have homodimers and we have heterodimers. All right. And we talked about this, head to tail for heterodimers for metabolite, right? So they bind to the DNA. You don't have to know the ligands. You don't have to know all the ligands. You just have to know metabolite versus steroid. So this is kind of an interesting side note. And it looks like we have 1 minute. So I'm gonna I'm gonna pick up with this when we start back on Wednesday, and then I'm gonna have some things for us to do in class. That'll be good review things. Because that's the end, right? Yes, that is the very last slide. Okay,
You're gonna be in your office for a minute?
yeah, I am.
I gotta go print home for
Yeah.
my literature.
Yeah, and if I'm not there, if I just so happen to get stuck with somebody, just write your name on the top and stick it in the folder on the door. That's fine. Okay, anybody on Zoom need anything? It's just so surprising I ended a minute early. Okay. Oh, good, thank you. The test is Friday, yes, the test is
I
Friday.
emailed you this morning. Can I drop off my lab report during your ultrasound?
Oh, that's fine.
Around like
That's fine.
2:30.
That's fine.
That's fine. Okay.
Yeah, absolutely.
Because I'm meeting with Rachel to talk about the conclusion because I feel like I'm not grasping it.
And you know where to turn it in on the door. If I'm not in my office, if I
Okay.
am in my office, you can just hand it to me. But if I'm not there, there's a little black like
And just stick it in
just
there.
stick it in there.
Okay, perfect.
Yeah.
Here's mine.
You have your as you flip through. Oh, you have your reference. Okay, awesome.
Yeah, I
Some people
just,
didn't, and okay, so that's cool.
yep, and I put in my conclusion that this was at the end.
Perfect. I love it. Test is Friday. Okay, and I'm still. Okay, let me end the Zoom if nobody has any questions.